So I think, yeah, everything seems to be working now. Hi, hello, and welcome. Microbehunter here. Welcome again to another Saturday live stream. Yeah, a lot of people joined in this week. Yeah, I'm so always happy and glad that so many people are showing interest in microscopy. Audio is good. I see. I see that a lot of people are already commenting today. Yeah, a short disclaimer at the beginning. I don't know if you're able to hear it, but I've got a pretty bad cold. I've got a runny nose. My voice is a little bit not so good today. I've got a slight headache as well. So occasionally I have to probably take some tissue paper and clean my nose. I hope you don't mind so much about that. Well, what am I going to show you today? Well, look what I got here. I got some 14 antique microscope slides which I bought over eBay. And I did not look at them yet. I got them just recently, a few days ago. I unpacked them, but I deliberately did not observe them yet under the microscope because I said I want to wait for that and I would like to do that together with you. So yes, it's going to be completely new to me, just like to you. And maybe some of them look nice, maybe some of them look pretty bad. I don't know. Yeah, it's going to be a little bit of a surprise for all of us. Back in February, beginning of February 7th, I think, I already showed you some antique microscope slides. Here they are. These were the ones that I showed you approximately 3 months ago. And if you are interested in watching that video as well, of course, you can find it yeah, in the live tab. You can see all of the live streams of the previous months. So usually at the beginning of this live stream, I spend maybe about 3 minutes or so just to say hello all around the world and to read a little bit in the chat. The reason is because it does take some time, a couple of minutes for people actually to join into the live stream, okay? So usually I use this time to say hello. So it is hello from Germany. Okay, sound is good. Hello from Connecticut, the United States. Okay, and there is already a comment from Marie here who likes the trailer at the beginning of this live stream. I have this music and the trailer running. Yeah, and just wants to say that many people actually ask about the nice blue background that my microscopic images have and videos have. And that is due to a specific contrasting technique that my microscope uses. It's called DIC. And this allows me to change the color. It uses polarized light, pretty fancy technology. Yeah, but I'm just, just to clarify this. From Montreal, Canada, Northern Germany, Belgium, Germany again, the Netherlands, Vietnam. Okay, yes, lots of people today. And yeah, from Switzerland, yeah, from Germany, from the United States, from Rhode Island. Sound is good. Thank you. That's important because once I actually started to talk and nobody could hear me. Yeah, clean your nose and put it on the slide. Actually, I have already done that before. What you're able to see is you're able to see a whole bunch of epithelial cells. So, yeah. Yeah, I'm gonna feel better soon, okay? So, how old are these slides? Okay, that is an interesting point. And it depends a little bit, but some of these slides actually go back to the 1920s that I have. And I know that because some of the slides actually carry the name of the company or the person who made those. And then if you do a little bit of research, and as a matter of fact, the webpage Microscopy UK, they have a web page where actually you are able to see they've researched all of the different microscope makers, and I don't know if I'm able to actually show this to you now. Just a second. No, that's not the one. I'm trying to maybe show this to you, but I'm not sure. Yeah, there it's if you Google microscopyuk.org. .uk, then essentially you're able to yeah, I'm not able to show you the window now. I don't know why this doesn't work. It doesn't matter so much. Yeah, but in any case, some of those slides can be around 100 years old. Yeah. Yeah. Yeah, and of course, if you want to have if you have a specific question to me, then please put an @Oliver or @Microbehunter at the beginning, so that I know that I'm able to respond to it. Okay. How do I know that they're 100 years old? Well, I know that to a certain, some of them might be that old because if you actually take out this here, then there is sometimes the name of the person written on here, and then you do a little bit of research, and then I found out that some of the slides actually go back to the 1920s. I'm not sure about specifically this one over here, okay? Arthur J. Doherty. And if you Google him, then you're actually able to see when the company actually was in operation and when they actually sold slides. Yeah, so that's a little bit the thing, and I already have to interrupt myself a little bit for a minute. Excuse me. So now I feel a bit better again. Okay, so to give you a little bit. Yeah, so what sort of mounting medium did they use back then? I don't know exactly which mounting medium they used here, but Canada balsam was very popular, and actually Canada balsam is also known to have a very good long-term viability, or how do you call this? It's able to store for a very long term. Okay, Canada balsam, not so common anymore because, it does require quite an extensive sample treatment. So I got those slides over eBay, and these are essentially the slides here. So a total of 14 slides which I bought. The price range was from around 10 British pounds to around, for a set, to around 20 British pounds for a set. Some of them actually, these ones here I did not auction. I just basically bought them directly because I did not want to wait, hoping that I'm able to actually get the best bid. So I simply chose those that I could buy directly. Maybe these are not necessarily the best ones. But in any case, we're gonna have a look, okay? Was kann man tun, Luftbläschen in selbstgemachten Dauerpräparaten zu vermeiden? So the question in German is what can you do to prevent air bubbles in homemade permanent mounts? What do you do to prevent air bubbles? There are several techniques, and one possibility could be, a very quick one, is to put the mounting medium and also the slide into a vacuum chamber. You call this in German, you call this Wasserstrahlpumpe, or a water pump, it's called. And water pump is not the right thing. You connect it and you turn on the tap water, and this creates a vacuum. Right, or maybe you have some kind of a possibility to actually also create your own vacuum chambers. And some time ago I actually presented one because one of my viewers actually also made a vacuum chamber. Okay, using a pump, and not a pump but a syringe, a large syringe. You can actually make your own vacuum chambers as well. Okay. Why do you have those slides, and what do you hope to achieve looking at them? So the question is you know what, why not I just show you some of those slides here? The question is why did I even buy them in the first place? Okay, that's a little bit the question, and there are really two answers to this. The very trivial answer would be I bought them because I want to show them to you, right? I want to have some content for the live stream. That is a very not a very convincing maybe answer. And the other reason is because I do personally like a little bit the history that's connected to those slides. It's a little bit like collecting the slides. Now I do have to say the following, that the specimen themselves are really not that special. I mean, that's not what makes them so. Because I mean, what does it say here? The tongue of I don't know. Yeah, the tongue of a housefly. Yeah. Honestly, this is nothing really special. I mean, you pretty much any type of modern slide set that you buy over Amazon will contain housefly parts, insect parts, right? So the actual specimen itself is really not that special in most cases. I do have one over here that I kind of consider pretty cool. Yeah, this one over here. But why I like them is because a little bit of the artistic value maybe, and also not so much artistic even, but maybe a little bit also of the history that's connected to it. It's a little bit of a collecting thing. And I know that some people out there actually do like to collect microscope slides. I mean, I just want to show this. I actually made a separate video of this. Something like this slide over here is something that you're probably not going to get so easily. Yeah, here's the cross section of the tail of a monkey. Okay, so it's a monkey's tail cross section. It's something that you normally wouldn't get. And it's a very nice specimen. I even made a separate video of this, right? So in most cases, I collect those slides and I want to show them to you simply out of interest. But there's also this personal interest that I have. Okay, so that's a little bit. If you're kind of new to this channel. Then you might be kind of wondering what's kind of the overall purpose of this YouTube channel that I'm doing here, not only of this live stream, but generally of the videos. And that is a little bit of a background info before I show you the slides. Is that in my real life, so to say, I'm a biology teacher. I've been a biology teacher now for 25 years. And I'm a trained microbiologist, so I studied research microbiology at the university and got a master's degree, and then later on I decided to go into teaching. And I realized that teaching in a school, even though generally I think I can say that the students generally, I guess, like my biology lessons, I sometimes do feel that the syllabus that we have to teach and the way that we teach sometimes constrains us a little bit. I'm standing on a chalkboard and I'm trying to explain abstract concepts, and the students have to understand that. Cell divisions, the parts of a cell, I don't know, biochemistry, all of the sciencey stuff, right? And sometimes I get the impression that the real biology, and real under quotation marks now, the real biology becomes lost a little bit. It's the observation of nature, right? My love for nature, my love for observing, of simply discovering things under the microscope. And even for me as a teacher, sometimes this falls short a little bit. So, and I try with this type of YouTube channel and the videos that I'm making, I'm trying to also promote that aspect of biology a little bit. Okay, so that's a little bit the philosophical background. Okay, so I've been talking quite a bit now. But I'm always checking also the comments here. I just prepared my first few permanent slides with Elmer's PVA glue. I was wondering if pressing the slide is a common technique, and I designed a 3D printed slide press. Yes, it is possible. So the question is now, is when making a permanent mount like this over here, is it a good idea to actually press it flat? And I say, why not if the specimen is sufficiently thin? If you'd make a whole mount, let's say a small, I don't know, fly maybe, or insect, and you kind of press it flat, then of course you might be damaging the specimen. So my suggestion is always ask yourself, are you doing any damage? But generally I think that by pressing it does have certain advantages as well, because you actually make sure that the object is in the same plane of focus. Yeah. So a little bit of pressure, I think, doesn't hurt, but do be aware that you might start to introduce some tensions into the cover glass as well. Okay. Yeah. So those slides here are sealed somehow. Yeah, that's the ring that you see over here. That's correct. These slides are ringed. I also made videos and live streams about slide ringing. And the ringing has actually several functions here. And it is not always necessary. It depends actually on the mounting medium used. And sometimes it is necessary. Now, if glycerin jelly has been used, or glycerol gelatin has been used, then it is absolutely necessary to ring it, because it does not dry out completely, and putting a ring here prevents moisture from entering and escaping, and it also makes sure that other, let's say, fungal spores and so on, contaminants, are not able to reach the glycerol jelly. Okay, so it's important for that. Number two, it also stabilizes the cover glass a little bit. And number three, some people actually use this also for decorative purposes. And I again would like to show you the one from, ones that I have over here. Yeah. Here, for example, you can actually see that the ring here has also been used for branding purposes. Okay, I have to focus here a little bit, right? Now you see that it's multicolored even, so this is actually also used for for branding and for decorative purposes as well. Okay, so but it does actually for ringing you can also use nail polish. Okay, so so what I'm going to do now is this is now where the real thing starts. I'm going to take them one by one now, and I'm going to put them under the microscope, and and honestly, I really don't know what to expect. Okay, so maybe we're going to be lucky, maybe we're not going to be so lucky. I don't know. Okay, so this is a first for all of us. Yeah, I deliberately did not look at them before. So let's take out the first one over here. Okay, so this one is it says here 2 antique microscope slides, the transverse section of a linden tree and gold rayed lily. So which one is this one over here? Yeah, that's the lily. Okay, lilium. It's the anther, transverse section of the anther. Okay, here there's even some more information here. It's stained in carmine, in balsam and benzol. I suppose they refer to Canada balsam. Okay, and benzol. I don't know what this is. Seems to be some kind of a solvent, maybe for preserving it better. Okay, so this is the transverse section of an anther stained with carmine. Carmine, this reminds me, carmine actually is able to stain chromosomes. So I don't know if we're able to see cell divisions. That would be really cool. But I have not very high expectations yet. So this is going to be the first time that this slide has been seen by a lot of people. Yeah, and this is, here we go. Okay, yes, it's nicely stained, of course, completely out of focus. Here we go, and we see the pollen grains. Okay, what I'm not going to do is I'm not going to be cleaning the slide because I do not want to apply any pressure. And I just realized that you are not able to see the... Yeah, now you're able to see it. Okay? So you see the slide up there, right? And you can also see that I'm now using the 4x 4 times objective. Yeah, and let's move up. And I'm going to remove the filter so we're actually seeing it in bright field, just like you would be able to see it. Okay, actually this is a pretty nice one. This is a, look, you see the pollen? I mean, yeah, you see the individual cells here. Okay, I'm gonna show, I'm gonna, you know what? That's my biology teacher disease, as you call it. I always have to explain. I like to hear myself talking a lot, but in case you don't know, that the anther, that is the male part of a flower, which produces the pollen, okay? So these here, those are red stained grains that you see here, these are the pollen grains, and you see the individual cells, and the little dots that you see here, these are the nuclei of the cells, okay? So each cell should have a nucleus, and if you see a cell that does not have a nucleus, then this is because the nucleus has been cut away, because the cell is a three-dimensional structure, right? And if the nucleus is here on top and you're just taking a section, cross-section below, then of course you're not able to see the nucleus, okay? But I'm actually, I'm actually quite happy with that, but I'm going to now zoom in more. Of course, I always have to go up with the, I've got a commercial slide somewhere where actually I could see, I was very lucky, I could actually see some dividing cells as well. You could actually see the chromosomes, but that is a little bit a question of luck. Actually, this is actually quite a nice specimen because it is shows the whole structure. And this basically means what I'm going to probably do is I'm going to stitch it together, right? I'm going to make a picture. So what you can do, and that's something that you might also want to try, is you take a video by simply going across like this, and going down and scanning it like this, kind of, right? And then you can use a software to actually create a picture, a stitched image. Yeah, so there's certain panorama software tools that are able to do that. But the reason why I'm looking here is because I was kind of hoping to maybe see, a couple of chromosomes. You see, these here, all of those dots here, these are of course the nuclei, yeah? And when the cell divides and the nucleus disappears, then you're able to see the chromosomes. This was kind of a little bit my hope. And for those of you who are a little bit new to biology and to things like this, yeah, of course, these here are on the outside here. These are the cell walls, right? Yeah, and carmine, actually, the red pigment is food coloring, by the way, right? They often also very often use carmine, and it likes to stain certain parts of the cell, especially also DNA. Yeah? And down here, that is again, yeah, these are the pollen. Oh wow, look at this. I wonder. You see, a pollen when it pollinates the female part of flower, it will form one pollen grain will form two sperm cells, and maybe these are the two nuclei, each one of them forming a separate sperm cell. I don't know. Okay. Does a single flower both have male and female parts? It depends on the flower. Okay, that's the simple answer. There are all combinations are possible. There are flowers that have both male and female parts, but generally self-pollination is not something that the flower always wants to do. So for this reason, sometimes to keep it separate, to prevent the pollen of the same flower pollinating itself, sometimes the pollen, the male and the female parts of the flower, they start to mature at different times, so that there is still a separation of time, yeah, so that self-pollination is reduced. Yeah? And then there are also other cases where you've got male and female flowers separate. So nature is very diverse, and if you look hard enough, you're probably going to find pretty much every combination. Right? Which microscope is this? I have an Olympus microscope, right? Yeah, it's and I've got a camera connected, and the camera obviously I have a camera connected, and the camera I've connected over HDMI to give me a live feed. And so I'm not using a microscope camera, but I'm using an Olympus. An Olympus camera. No, a Canon camera. My mistake. I'm using a Canon camera which I connected over HDMI, and then I put it into the computer. Okay. Can you go 60 times? Yes, I got to be careful because the distance is already very small, okay? So I have to be really careful. I have to see if I'm not crashing into it, but it seems to work. Yes, it works. I always have to turn up a little bit, and I'm going to open the condenser a little bit. Yeah, so this is now 60 times. Yeah. Yeah, and but I'm not going to go higher, okay? Because the next objective, the white one that you see over here with the white ring, yeah, is an oil immersion objective, and I'm definitely not going to put immersion oil on this microscope slide. Definitely not. Yeah. Because it's going to destroy my slide, and it's also not needed. Yeah, in case you are wondering a little bit, so a little bit of technology again. And then I know I'm talking a lot. I want to show you all the other things. Look what I'm doing right now. Look at the 60 times objective. Look at this, and at the same time look at the image, because this objective. Look what I'm doing here has a ring. And I'm able to turn this ring. Can you see this? Yeah. And this ring actually is I don't see a huge difference now, but this ring is there to adjust the thickness of the cover glass, yeah, to get the best resolution. Yeah. You can actually sometimes even see that the clarity changes a little bit. Yeah. I don't think it's an aperture. Because there's a question, is this an objective with an aperture? I don't think it's an aperture, but it is some kind of I wouldn't even say focusing, but it corrects the thickness of the cover glass. And now you see that the setting is 0.17. That is the current thickness of the cover glass. However, this also depends a little bit on the thickness of the mounting medium that you have and all these things, right? So it's So that's actually a very nice specimen, considering the fact that this thing is I don't know, almost 100 years old maybe. Yeah, it's quite nice. Generally, what I feel is let's go down a little bit with the magnification again. Down with the brightness. Generally, what I feel is that some of the slides that were made by those commercial slide makers 100 years ago or so. are of sometimes significantly better quality than some of the modern slides that you get. Yeah. So for example, what I mean with that is if you look at the whole structure here, you're going to see it's not ripped apart or torn or anything, but the anther cross section is quite complete. Yeah, not quite complete, it is complete. Yeah, but I've seen some modern microscope slides where it's been cut a little bit sloppily, and then I don't know, it's torn or ripped, yeah, or there is a significant amount of dirt. I think we'll always find a little bit of dirt on a microscope slide, but sometimes it's quite quite a lot. Yeah, so I think quite nice. Yeah? Does water-based immersion medium with the correct refraction index exist? Would it be easier to clean? What you have is when you basically what you can use is there are so-called indeed water immersion objectives. So what you use is you just use regular water as an immersion medium, right? So there are these special objectives actually are available, but not with low-cost microscopes. You have these are pretty specialized objectives already. Yes, but water immersion objectives do exist. But because of the refractive index of water, I think it doesn't work quite as well as oil immersion. Yeah? But yes, it does exist. And I know of one company even which advertised their objectives also to be usable with water. So you're basically putting a drop of water directly on the microscope slide and you're rotating the objective into the water because it's easier to clean. Yeah, so and the small reduction in image quality that you get, you can forget about that. Yeah? So yeah, I'm quite happy with this one, actually, and maybe I'm actually going to make a picture of this. Okay, so I like to talk a lot. Let's turn the arrow off, and I'm quickly reading a few comments here. I'm pretty new to microscopy. What microscope would you recommend for beginners? That's one of the that's a difficult. Well, actually, my suggestion is, and I know that people disagree sometimes. Do not get yourself a toy microscope. Okay, but if you really, it depends a little. Look, there is no limit to the top, right? But generally, I recommend that while you shouldn't get yourself a toy microscope, do not spend too much at the beginning. Okay, you kind of as you because you see, if I were to if you were to put the slide under a fairly cheap microscope, it would not look so much different, the image quality. Yeah? So it is So this is the thing. So it doesn't really matter so much, I would say, but don't spend too much money and then kind of grow with the hobby. Yeah, a lot of it is a question of personal taste. If you're really serious about microscopy, get yourself a microscope with a condenser. Okay, so you will probably have to pay minimum of around €300 or $300. Okay, are you looking at the slides for the first time as well, or did you already have a glance? No, I did not have a glance. The only slide that I had a very short glance under the stereo microscope was one of the forams for foraminifera, but I only for 5 seconds or so, just to check if something was there. Right. So you know what? I'm going to move on. Okay, because otherwise, we're never going to get finished. No hurry, but still. Let's carefully take it away. So this was basically for those of you who joined in a little later. This was the slide. Okay, it is the transverse section of an anther of a lily. Yeah, and something I really like a lot is a little bit of information about what stain was used and what mounting medium was used. Yeah, so that's really something that So let's put it back. And what is the next one over here? Let's have a look at this one. This one is Tilia. That is some wood. It's a lime tree. Longitudinal section. LS means for longitudinal section of the stem. Stained logwood. Okay. It's also stained, also mounted in Canada balsam, I suppose. This looks pretty dark actually. not dark, but it looks pretty opaque. So I don't know if we're able to see a lot. Okay, so and normally specimens should be sufficiently transparent. Yeah. Was the stain color different 100 years ago? So basically the one that we just looked at, carmine, was also red. So it did not bleach. But it is indeed the possibility that some stains can bleach. Yeah, but I know because I've recently done some how do you say, some staining with carminoacetate, and it is also, yeah, it did not lose any color. So this is now some wood. Okay, let's go up a little bit here with the it's a long, look. Okay, that's a fairly thick cross section. So you see the lignin, these are it's the wood. And yeah, maybe you're able to see this. I'm turning on the where is don't I have the arrow here? Here is the arrow again. It could be that, do you see those little horizontal stripes? It could be that this is the so-called xylem. Xylem are those cells that actually transport the water upwards. Yeah, and let's go up yet more. Yeah, looks like it. Yeah. There seem to be, it's interesting, there seems to be a little bit, some bubbles here as well. Okay. Yeah, but, yeah, here it is. Here it is. This could be actually it. You actually see those horizontal lines here, right? Yeah. That's very typical of plant tissue, and you find this quite commonly in plants. And these are basically then the cells that transport water, and those ring-shaped structures actually make sure that the cell is kept open for transporting water. It's a little bit like over here in the windpipe that we have. We've got also those cartilage rings to prevent our windpipe, to keep the windpipe open. And we have the same thing here also when a plant, when there's a water stream inside a plant, because of the movement, there is always the danger that the plant cell walls would then kind of collapse due to the lower pressure. And for this reason, there are these strengthening rings. So that's called xylem, these cells here. Right, and it's you find this quite commonly. But I think this section here looks fairly thick. Okay, so I actually have seen, here very nicely visible the xylem tissue. Yeah, so if you happen to see some spirally ring-shaped long tubes, like I don't know, do you know those slinkies? These are those spirals that kind of go down the stairs, the toys, right? It kind of looks a little bit like that. And if you see this under the microscope, then you know that you're actually seeing xylem tissue, and those xylem rings are actually thickenings of the cell wall of the plant. Oop, I bumped now the camera, and yeah. So I'm just checking again. Do 100 times dry objectives exist? Let's put it this way. If you don't put mounting medium on it, not mounting medium, immersion oil on it, then you have a dry objective. And I have actually, yes, I think I've seen something like this, but you do not get a better image quality. You see, what is important, that's really, do you see this number over here? Like the 0.50? That's the numerical aperture, and that actually determines the resolution. And if you have now an objective of, let's say, 100 times objectives with a numerical aperture of 0.65, then this is less than a 100 times objective with a numerical aperture of 1.25. Right? So this means even though you have the magnification of the dry objective, you don't get the image quality. So there is actually no point in magnifying something where you do not get the clarity, right? So while I've seen that occasionally these objectives do exist, there is really no point, because then you just might use a 40 times or 60 times objective with the same numerical aperture, right? And this gives you even a better image quality or the same image quality of clarity, right? So this is a little bit the issue here. While 100 times dry objectives, I don't know, I've seen this somewhere, one company making those ones, but actually there is no point. Yeah, just use a lower magnifying objective with the same or higher numerical aperture, and you're going to get a better image quality. Yeah? Very nice. Here, the xylem, beautiful. Look at this. Do you see those dots here? Could this be actually? Could these be the openings in the cell wall which allows the water to pass out sideways? I don't know. Maybe some of you are more into botany than I am. Yeah. So diffraction limited. That's the point. Yeah. There is very often or sometimes a little bit the misunderstanding where people think the higher the magnification automatically the more you see. But that is not always the case. There is no point in. It's almost like taking a mobile phone and you zooming in into a picture, right? Sooner or later, you're not. It's just blurry, right? And it's the same with microscope objectives. There's no point in zooming in if you do not also get the resolution, right? And there is a certain physical limit. Yeah. So that is aberration gets bad too. Yeah. So that's basically color aberrations. Chromatic aberrations and so on. Actually, that's kind of, So that's basically the wood, right? Seen from the side. Seen from the side. Yeah, you know what? Let's go on. What time is it? It's already how many minutes? I'm already talking half an hour, and I've only looked at 2 slides. I actually want to look at a few more. Okay, but some interesting outgrowths here. Yeah. So again, this is let's go down with the, just for the fun of it, I'm going to switch on now DIC, but there is not a huge difference. It's a little bit darker, everything. Let me see, I have to get the correct filter setting here as well. Let me see. You see the color change in the background? Yeah, all the way on the left is very limited. Yeah, so does not seem to change a lot. So there's no point. Let's just stay with bright field, I would say. Yeah. Okay. Let's move on. So these were basically the 2 quality slides from a well-known microscope slide maker. Already back, if you just joined in, back in February, if you're interested, I already made a live stream where I showed you several slides and also of the same slide maker. So I already knew that his slides are pretty good. And now I've got, I don't know, what does it say here? The next one, 4 antique microscope slides by J.W. Bond. It is Sting, tongue, foot, and guano. No, actually, I'm going to show you. It's actually Infusoria, or it's written on it. So these are those slides. These are the so-called the paper-wrapped slides. And unfortunately, I've seen that one of them actually is broken over here. It's only held together by the paper. So we got to be a little careful here. Okay. So these are completely paper wrapped slides. And this one over here is the, what does it say? Let's focus. Sting of a bee. Okay. It says here, it's handwritten over here. And again, you have to excuse me for 5 seconds because I have to clean my nose again. So, yes, I'm saying a very authentic thing here. So, what are those lines on the slide? Which lines are you referring to this slide here? I don't know. Okay. There is a question here. Is this which lines? I don't know exactly which lines you're referring to, and I think there must be written. Yeah, somewhere there is no brand name over here. You know what? Let's put it under the microscope. Again, this stinger of a bee is nothing super original because it's one of those classics. I've even prepared stingers of bees and wasps and so on. Okay, so let's go down with the magnification first, simply to find it. It's way too bright. Here we go. Ooh, okay. The mounting medium already suffered a little bit here. You see those bubbles and so on? Yeah, so. But what we have here is the stinger of a bee. So this is now with a 4 times. If you're kind of wondering why this is in a circle, okay, just look what I'm doing right now. That's because of the condenser. You can actually swing out the condenser if you want to. Okay, where is this? Yeah, so you can swing out the condenser like this. Okay, and then you're actually getting a full view, but the brightness is not as high. Yeah, and so, and here, that's, yeah. So let's see. Yeah. This is the stinger of a bee. Let's see what we're able to see here. I think this seems to be seen from the side. Yes, that's the stinger. It's a little bit, it looks a little bit broken. But there is something that I actually want to show you. I did also make some videos about this before. Let's go up yet further. There are, I see, I should have adjusted the microscope to always go up with the brightness as well. Yes, the stinger is a bit broken, doesn't matter. But the thing that I want to show you is still visible. If you look at the stinger, then you might see actually those tiny, teeth-like structures here, right? Yeah, so these are kind of hooks. And this basically means when the stinger goes into your skin, and then there is this, in the center here you have a tube where the poison is squeezed out. And then the stinger will stay in the skin. Okay, because those hooks make it difficult for the stinger to be pulled out. And this actually means the bee will die. Because on the other end over here, so there is a tube on the inside for directing the poison. And there over here on the other side, not so well visible. I think this one over here must be then the gland producing the poison. I've seen actually from a top view. My other slide has it from the top view, and it's a little bit better visible. Okay, but in any case. If you put a stinger of a wasp, for example, under the microscope, then you're going to see it looks very different. It's very smooth. So I'm going to quickly check again. Yeah. If I did not look. Have you ever thought about examining nasal discharge for a stream? After all, everybody has it. You know what? Maybe I'm gonna try that. Okay, it's mucus, and I don't think that there's gonna be so much in the mucus because it's produced by the glands, right? But if you kind of blow your nose, then there will always be epithelial cells coming along as well. What I've done once is I coughed up something and then put it under the microscope slide, and I saw a lot of cells. Yeah, so it's just basically your own epithelial cells that come up with it, and then kind of that's the thing that you're looking at. Yeah, so this is over here because somebody asked me what about pressing? What about what happens when you press it? And this is actually something that might happen. You see over here on the top that the specimen is ripped. This can actually be when you apply too much pressure. Then of course the specimens can deform and rip. Yeah. These things here actually are artifacts as well, where it looks like that the cover glass kind of started to slowly separate from the mounting medium. Yeah, and then you have these bubbles. Yeah. This is actually normally when you have bubbles, then they look more round, but sometimes, but this does seem to be some kind of a separation. Okay. Again, as I said before, those specimens as such are really nothing special. I mean, stingers of a bee you find in so many commercial microscope slides, right? The reason why I am interested in those specifically is because they were made quite some time ago. Okay, so let's put the arrow again here. All of the here. Yeah. Here again a little bit ripped. That's interesting. What is that? I don't know. Some openings in the exoskeleton. Yeah. As a matter of fact, I've seen a bee stinger where actually the two parts of the sting, there are actually two parts of the stinger, and apparently what they do is they kind of move back and forth a little bit like a saw, and that's how they're able to go into the skin more easily. And then you have those tiny little hooks here that actually prevent the stinger from coming out again. Yeah. The question is why is the slide decorated? So in other words, why is this slide here? It's covered in paper, and you see that these ornaments and it's a decoration on it. The paper itself does make the slide more stable in the sense that it doesn't break quite as easily. A little bit ironic because this one still has a crack on the inside. That's why one corner is a little bit loose. That's one thing. And number two is that those microscope slides used those slide labels also for branding purposes. So these are sometimes commercial slides that were made some time ago, and they were actually used for distinction and recognition and for branding purposes. And these days, they're used also for collector's items as well. So there is not always a rational, objective reason behind it, but those microscope slide makers, it was very evident at that time they did not make those slides only for scientific and research purposes, but actually also because, as a form of artistic form of expression, I would say. That's what I would probably say. The mounting medium shrinks, leaving spaces. That could be indeed one thing. Normally, a good mounting medium should not shrink. However, what can happen is the following. And this is something that I've also experienced in some of my slides. Is the following. If the specimen is not completely dry, okay, there's a little bit of moisture still in there, then it can be that bacteria start to decompose the organic material that's in there. And when bacteria start to decompose material, sometimes they produce gases like carbon dioxide, or in the case of anaerobic bacteria, even methane gas. Carbon dioxide gas, methane gas, hydrogen gas maybe. And these gas decomposition gases might also start to build up and will also start to form bubbles. I've actually seen this, that in one slide there were actually, which was stable for some time, and all of a sudden I could actually see that gases started to build up around the specimen. Right, and there was actually no connection to the outside, right? So where did the gases come from? And I think that these could be decomposition gases. Yeah. A pun. No objective reason. There is no objective reason for actually paper wrapping those slides. So let's move on. Let's move on a little bit. I'm going to now put the next one. So that was the stinger of a bee. Let's put now the next one under. There's by the way, there is, you see, that's unfortunate. There is this corner has cracked. Yeah. It wasn't me who did that, but this can actually happen. Maybe it fell down some time ago. I'm not going to glue it or anything. I'm just going to leave it in its original state. However, there is an interesting thing. If you just look at the thickness here, you're going to see that this one over here, I had to put down here because it wouldn't fit into the other slots because it's significantly thicker. So at that time, actually the consistency of the thickness of the slides was not the same either. So let's have a look at this one over here. Tongue of a housefly, okay? Yeah, tongue of a housefly. So scope again. Let's start again at the low. You always start at low power. Yeah. It's again out of focus. Excuse me. Okay. Where is the tongue? Yeah. Here, this is the tongue. Okay. And these are the mouth parts, so to say. Really? Okay, the one that I've actually seen in another one looked a little different. Okay, normally there is some kind of a, interesting, okay. So, let's have a look. Okay, so these are the mouth parts. So, plenty of, interesting, okay. Here we go. Seem to be. Maybe that's the one here. Seems to be a little bit. A few parts seem to be broken. It doesn't matter. Here. Yeah. I'm just a little confused now because it looks so different to the other one that I have. Yeah, it's also something quite fascinating, not fascinating, but something that I could observe already several times. Um, Look at this. There seems to be, if you look at the mounting medium, here it's clear, and over here it's a little bit more yellowish. It almost looks like some of the stain, not stain, but some of the pigment kind of leached out into the mounting medium. Yeah, here it was cut off, evidently. And what is this here? This is a long thing here. Wow, okay. A long tube, and I don't know what it is. Okay. Yeah, so this is a fly. When preparing insects, one of the big challenges, of course, is that the exoskeleton of the insect is usually very dark. Therefore, often you are not able to see a lot. So for this reason, you have to bleach the insect, the exoskeleton. You can do that by putting it into a solution of an alkaline solution, like for example potassium hydroxide has been used quite a lot. This also softens the exoskeleton and makes it easier to prepare. Yeah. But I don't know if this one has been bleached because over here it's still very dark. And there are also certain mounting media that have a natural clearing action, and they will naturally clear it over time. Yeah. So that's also possible. So again, I have to clean my nose. Yeah, so let's go down again a little bit with the magnification. Okay, so here we go. Yeah, so this seems to be a little bit, this specimen seems to be a little bit dislocated. Yeah, because it almost appears like that this part over here obviously should belong to over there, maybe extending outwards, and maybe this kind of bent over a little bit during the preparation. Okay, so let's move on. I'm occasionally looking for some comments again. So this one was this, and the next one. What's the next one? Oh yeah, this slide here is also a little bit damaged. Foot off. I don't know. I'm trying to focus this here. I cannot read this. Okay, maybe it just says foot. Okay, I don't know, some kind of a foot. Yeah. Yeah, there's a question here. When I see microplastic is always bright blue, I can confirm. Is this because of the refraction index of the plastic? Probably not the refractive index. But indeed, there are many blue synthetic fibers out there. I don't know why it's always blue, but the refractive index, changes in the refractive index should not impact the color so much, unless you're using some kind of polarized light. So this is some, here, that's already a foot. And a foot. I don't know which one, but it's a foot. Fly feet look also like this a little bit. Yeah, that's either, I don't know, the claw or whatever. And you can count the segments. And here we go. Yeah, this looks quite okay. And let's try to zoom in a bit. Let's get the arrow out of the way again. Occasionally, it depends a little bit on the specimen, and how it was prepared. Occasionally, you could even see the muscles inside, which are actually moving the legs. Let's try it like this. Let's try it like this. Let's try DIC. That's now the special contrasting. Look how this actually, this region starts to shine up. So you see that the polarized light somehow impacts the whole thing. Okay. perhaps about microplastics, perhaps the blue stand out. Okay, this could be indeed that the blue ones actually are more visible because you don't have blue color so much in nature, right? And the other fibers you kind of tend to ignore. Foot of Fiora. Thank you, thank you. What is a? Can you tell me? Maybe you can research a little bit. Which insect this is? Okay, this would be quite interesting. Lots of a brittle star. Okay. Echinoderm. Echinoderm. Echinoderms are actually starfish and sea cucumbers. Actually, these are not insects. Okay. Aren't fly body parts more opaque than this? And yes, that is correct. So normally, generally insect parts are very dark. And the question is now, why is this so much brighter? And that's, again, I think because these insect parts have been bleached. You have to chemically treat them to make them more transparent. Otherwise, you're not able to see anything. And wow, this looks pretty interesting. Yeah, so yeah, again here a little bit of an issue with that you see that the mounting media has not properly, there seems to be some bubble formation here. Yeah. So yeah, but as I said before, bleaching is absolutely necessary. So I've heard that you can, well, of course, if you are using the mounting medium Uperol, which I use a lot, difficult to obtain. Some people in the comments section have complained a little bit that they were not able to buy it because the companies would not send them to private people. That's always a problem, that many chemical companies do not want to send certain substances to private individuals. Even if the substances are not so poisonous, they generally don't sell to private individuals. So this means that some of the mounting media and can be difficult to obtain. If not, then bleaching is possible with potassium hydroxide. I do not know the concentration now, but I know that there are also other YouTubers out there who are really spending quite a lot of time preparing insects and also spiders and so on. And I've seen some very interesting YouTube videos. Okay. Is it a fly foot? Could be a Drosophila foot. Yeah, but again, it's labeled. Let's take it out here. And it is labeled here. And foot of I cannot read this. Opticon, I don't know. Yeah. So this is how it's labeled. Yeah? And over here on the sheet of paper, on the bottom of eBay, it just basically said foot. Okay. So the next one is something I really want to know because it actually says here the following. It says here guana or guano, and the first word here seems to be infusoria. And infusorian was actually an old term that was used, generally back in the day when they made so-called hay infusions, something where you got to be really careful because you're really growing a lot of bacteria as well. So they took some organic material, some hay, some grass, added some water to it, and allowed the microorganisms to grow. And what they found, basically they called infusoria. All of those protists that they found. Of course, you have to add some pond water as well to inoculate this. And it's a general term for water microorganisms. And guano, actually, that is used also as a fertilizer. And that is actually sand mixed with the excrements of certain birds. It's called guano, and that is actually a very good fertilizer. Yeah, so I'm a little bit, I don't really know what to expect here. And just putting it here, I don't see anything here either. So I don't know what the, yeah, what to expect here. So let's have a look whether these are indeed what this actually is. So let's remove this here. I'm not able to see anything yet. Ah, yeah. Maybe it's the sand, the guano sand. I don't know. Oh, it's that's there is something here. Oh, this looks like they look like diatoms. These are diatom shells, the round marine diatom shells. So maybe it is guano sand. Oh, that's cool. Okay, that looks quite nice even. Strange question. I'm just going to read this here also while I'm kind of wondering here. When you cook meat that has been frozen, you see white foamy liquid going out of the meat. Is this cytoplasm because of broken cell walls from the frozen? These are probably what the white stuff that you see coming out. That's the same. Look, when you take raw egg and when you cook raw egg, it turns white. The liquid is actually a liquid, can be broken cytoplasm, doesn't even have to be, but it contains proteins, and those proteins in the heat, they start to denature, they start to form cross-links, and this looks white. So the white stuff coming out of when you heat the meat is basically the same as protein, right? It can be broken up cells, but that contains also protein. Just like cooking an egg, which also turns white. Okay, this is actually quite nice. This is just a second. I need to. These are marine diatom shells, and I know that guano sand usually contains the waste of birds and so on, but this is wow. Yeah, these are made of silica. Called frustules of diatoms. And these are marine diatoms, most likely because they contain a lot of round structures. Do they have, the thing is I turned off I took out the polarization element. So this is what we see now is bright field. So this actually must be the natural colors. I'm going to go up now to 40 times. Or maybe, yeah, and when I twist the filter, that's kind of interesting. It could be what could be. That's an interesting one because you actually do see. Now that's a speculation from my side. You do see some colors here, right? It could be because of the fine structure of those frustules here, and you see that there is this very tiny regular pattern that this causes the diffraction of light and an overlaying of the different wavelengths, and basically certain wavelengths cancel out, and this might actually result in a color. Okay, that actually you have some cancellation of certain wavelengths because of this irregular pattern. Yeah. So this is my guess why you actually have those colors here. Yeah, different wavelengths are diffracted differently by those tiny patterns here, and then they cancel out, and then destructive interference, and you might get those colors. Yeah. So that's actually a nice surprise. I didn't expect this. Because it says here like Infusorien in German, this term was used quite commonly for water samples as a general term for water microorganisms. Yeah, this is actually quite nice. I'm really not happy with this one because I don't have a slide with that density and with so many of them actually. Yeah. Yeah, so Yeah, so Michael Kremer is saying the colors on diatoms are the fine holes here that seem to be causing some kind of a diffraction pattern. Yeah. You know what I'm going to do just for the fun of it? I'm going to now switch on DIC. Let's see if this, now I'm actually using the polarization effect of DIC. And of course you see that there, ah, look at this. You can play around. So that's actually quite fun. Ah, look, yeah, they respond differently to the light. But you know, I'm just going to stay with bright field because that's actually the thing that most of you will be seeing. Yeah? The diatoms show the food chain. The diatoms were eaten by copepods and other plankton, which was eaten by small fish, which were eaten by larger fish, and then by the seabirds. Well, let's put it this way: the diatoms are so-called referred to as belong to the so-called the phytoplankton, which basically they do photosynthesis. What you see over here are just the silica shells. Oops, now I bumped it. Okay, because when they are alive still, no, I lost it. Then they are either green or sometimes they also have a brownish color. So again, I've got a cold. Yeah, and we have somebody else joining in here from Altmünster in Austria. Yeah, hi, I know you. Huh? And if you just joined in, we're looking right now at very old microscope slides, which are bought over eBay. And I did not look at them. And those structures that you see here are so-called the diatom shells or diatom frustules. And they basically are made of silica, and these are basically marine plankton. Yeah, and they have those nice patterns. Okay, so let's move on because I still have a few slides to show you. As a matter of fact, we're not even halfway through, and it's over 1 hour. But I'm not going to rush. If I don't finish all of those, you know what? Then I'll just continue next week. Okay, so I'm going to take out the slide again, and let's take it out. So this was actually the one that let's go again to the desk here. Yeah, you see actually, yeah, infusorien, guana, yeah, diatom slides, diatom slide, a nice diatom slide. Actually, I think it's a little bit mislabeled. Yeah, so this is, yeah, so. And if you just joined in, those slides are around 100 years old. I bought them, yeah, over eBay, and they were made by different people, yeah, a long time ago, sometimes companies. So I'm going to go on over here. And one of the things that I noticed with those slides here, even though they were from the same manufacturer, is that the thickness of the slide was very different. So this one over here is very thin, and so this actually shows that there was a manufacturing inconsistency at that time. So I was not able to find out a lot of about this company over here. Entomology basically is the study of insects, head and mouth parts of a female gnat. Okay, so it's an insect, and yeah, let's have a look at this. So something I consider quite interesting is, just a side note, that were a lot of high-quality British microscope slide makers. I don't know why this is the case, but this seemed to have, especially Victorian slides from that time, it seemed to have a pretty strong tradition. Yeah, I have to go down with the light intensity. Yeah, so, and yeah, it was totally out of focus. Oh, nice. Look at this. Complete head, yeah, of a gnat. And look. Beautiful. This is also a pretty good one. It also shows all of the details here. Right head on view. The compound eyes are not visible. It's evidently a cross section. Look at this long mouth part here. Yeah. Look, it's quite fascinating here. And not G-N-A-T. It's written G-N-A-T. Yeah. What is this here? Was this maybe an antenna? Yeah. So that's quite good. This is definitely this one was definitely prepared because the head as a whole mount would be way too thick. Yeah. So and it must have been bleached as well as like many insects are, because otherwise it would be way too dark. Yeah. You can see the hexagonal structure of the eyes. Where can you see this? Maybe I'm a little bit too. Let's see if there's something left over of the compound eyes somewhere. So these things down here are not. These things here are not the eyes. Okay. Ah, yes, they're visible. Oh my gosh! Of course. Look. Ah, this was a very look. Okay, beautiful. Do you see those little things here? Okay, these are the individual elements of the eye, the so-called the ommatidia. So that means that the eye is pretty large here, and normally the eye is very dark. It's darkly pigmented. They have to be, in order to catch light, they have to be very dark, and it's extremely bright. So this actually is a sign that they have chemically treated the specimen to brighten it up. Okay. It's actually quite nice. Yeah, so that is basically, those little elements, bumps that you see here, these are the individual eyes, not eyes, ommatidia as it's called. And that's the whole thing here, that's one of the compound eyes. Okay. This is, this has definitely been bleached, because normally when you put it directly under the microscope, the eyes are very dark. Okay. I'm happy. Those slides, those old slides, those folks really put a lot of love and time into making those. Yeah. So this was evident. You kind of feel a little bit that the people who made those slides, they were kind of proud of their work, because it's not. It does take a little bit of time to make these things. Yeah. So that is a beautiful specimen. I'm very happy. Are ommatidia similar to eye cells? I think you are referring to the photoreceptors in the human eye. Well, they have a similar function in that they're light sensitive, but the functioning is very, very different. Yeah? Those in the... Oh, I bumped again. See? What did I do now? Okay. I have to close the condenser. Here we go again. I have to be a little more careful. Those individual elements that you see here, the ommatidia, what they have is these are long tubes. Yeah, they do contain something like a lens at the top, and on the bottom, on the other side of the tube, they contain a photosensitive nerve cell. Yeah, so the light actually is directed downwards. So it functions quite different. Well, there is a similarity in the way that it functions to the human eye or to the mammalian eye, in that it's of course light sensitive, but the design and the structure is totally different. Yeah, so yes, their craft and love for beauty results in our happiness. That's, by the way, it's one of the ways that the reasons why I make this YouTube channel. Actually, one of them, is because. It's not very scientific, really. Right? I'm not throwing around a lot of scientific terminology here. Like, for example, when you're listening to a zoology lecture, which I also had to attend, you had to know the different parts and all these things, right? It's getting very technical, because all of those parts that you see here actually have a name. But sometimes this kind of destroys a little bit the love of simply observing for the sake of observing. Yeah. For Hydra, use a big jam jar, take a pond sample with lots of weed, shake it up, and you can see them with the naked eye if present in the size of a jar. Yes, I also once actually collected a pond sample. I was very happy that I could actually also find a Hydra. Yeah. So what time is it? Okay, I'm going to give it 5 more minutes. So what I'm going to do now is I'm going to look at this next one. So... we're not able to finish all of the slides. It doesn't matter. Well, then we're just going to continue next time. And then I might show you some other things as well. Tongue of a bee. Okay, we had the stinger of a bee, now the tongue of a bee. So. And the scope of view again. Again, focus. Yeah, let's go down with the magnification. Here we go. That's the tongue of a bee. Okay, so that's the part that actually collects the nectar. Here we go again. Yeah, cool. I mean, honestly, the dissection work here, gee. Yeah. Look at all of those tiny hair. Yep, and in the center, of course, you see the tube. Yeah, where the nectar is being sucked up. Yeah. So yeah, I'm generally very happy with the quality of those slides. As again, for those of you who joined in, the slides that we're looking at are antique slides that were manufactured quite some time ago. And I got them over eBay, and even though the actual specimens themselves are not that rare, I mean, they're pretty much every slide box, modern slide box these days contains the tongue of a bee, the foot of an insect, whatever, right? Yeah. What are the hair of the tongue for it? Haha, yeah, that's a good one, right? The bees actually, those hair increase the surface area for sensing. So those hair, either are connected to chemoreceptors, basically for tasting, right? So they're able to sense different chemicals, and or they're able to also, when they touch something, then they move, and then this can also send a signal. Yeah. So actually, that's something very, very common in insects, that those hair are connected to nerve cells, so that the insect is able to sense its environment either chemically or by touch. Yeah. And those hair can especially be found there where essentially you need a large surface area for sensing. Yeah. And of course, it therefore should not surprise that the tongue of a bee contains so many hair. So a couple of suggestions. As a content suggestion, I recently bought an old HOC microscope. It's from around 1960. Maybe you have also an old microscope and could show it. Yes, actually I do. Well, not that old, but I do have a microscope from the 1970s, a CHA microscope, which I could actually show. But maybe it's kind of difficult for me to show it here because I don't have so much space on the table, but I do have a second YouTube channel called Microbehunter Microscopy, which is not quite, I'm not uploading quite as many videos as on here. And the other microscopy channel is a little bit more technical in nature, so usually I answer some technical questions like connecting cameras to microscope and optics and so on. And that would be actually a topic that would fit there. Okay. So I actually have my exams next Sunday, so we'll have to wait for the next Monday. Well, okay. Yeah. All of those videos are recorded anyway, and you can watch them anytime later on. What time is it? It's, you know what? There is one last thing that I do want to show or to look at. Just a second. And that would be the following, and I'm now going to switch on my stereo microscope because there is this one slide here, and this contains foraminifera. Yeah, foraminifera are the calcium carbonate shells of single-celled microorganisms that you sometimes find in sand. They almost look a little, sometimes they look like tiny snails, but they're not. Okay. And what I would like to do now is I would like to simply have a look at this under my stereo microscope. Yeah. Yeah. So I'm going to try this as well, maybe as a last sample for today. Okay, you see it's deactivated. The camera is always giving me some problems occasionally. Okay, let's zoom out. It's almost okay. I don't know if this is works so well, and I don't know if I'm able to see a lot. Ah, look, this is a little bit a pity. Look, the whole thing was a little bit. But just a second. This goes a little bit. Let's do it like this. Okay, I think that's better. And let's have a look. Let's zoom in a bit more. Yeah, you see the outer exposure of the camera is not the very best. This looks like sand. It's not. Let me see. It's not very clear. Not very clear. Okay, so I think I'm going to change microscopes again, and I don't know if I'm able to see more on the other one. But it is a little bit blurry. But I think if you're, looking around, then you might be able to see some of those tiny shells of the forearms. Let me have a look at the other scope. Let's remove this one here. And this is actually I was really hoping that I'd be able to see something here. It's nope. It doesn't work. Okay, this is a slide. You see it's completely dark, and I see that there is absolutely no light going through. They seem to have made the bottom side. Yeah, it's too dense. Okay, so unfortunately, look, it's so dense that maybe they even glued some paper on here that absolutely no light is able to go through. They actually, put it on paper. Look what they have done. That's an interesting way of preparing. So this slide cannot be viewed with a compound microscope. What you see over here is, let me put this away. You see here two rings. Actually, there's only one ring. Let me use something to point. I'm going to use just this here. You see over here, this ring that you see here, the inner ring, is actually not a ring. It's a black piece of paper. And they put the sand on top. And then they sealed it off again with the outer ring. And if you turn it around, you're able to see that there is actually the paper over here is white on the other side. So for this reason, I'm not able to look at this using my compound microscope, unfortunately. So you need a stereo microscope. It's the first time that I'm actually seeing that people have actually prepared a slide like this, that there is actually a paper background. It's interesting. I never see this. But this means, unfortunately, I'm not able to use my compound microscope to observe this. So why would they do this? They would do that to increase the contrast with the background. So if you are actually looking at it from the top, then you're able to see the white structures on a black background. Okay, that is the reason why they've done that. However, unfortunately, with my current setup, okay, if I put it on here, you can now see actually that there's the white sand on a black background. But unfortunately, maybe you have to go in a little bit more. Unfortunately, it's not clear enough to actually see the things very well. And sometimes if you look carefully, at the last time I've actually looked at this at forums like this was already quite a long time ago, and then you scan it around and then you're actually able to see some tiny little structures that look like snails or something like that, but they're not snails, but these are the shells, the calcium carbonate shells of those single-celled organisms. I just show you this one over here. So where is the arrow? So it could be maybe that this one over here could be an empty shell. You see that there's a hole in the center, right? but the current setup that I have is not clear enough for me to show you that to you. Yeah? So or these things, you could be maybe spiral. Yeah. So I'm going to try the following. I'm going to try to make some nice pictures of this so that we're able to and maybe enhance the contrast. Look, for the example, this one over here. Yeah, looks like it's a little bit like a it's pointed. It's like looks like a snail, but it's not. Yeah, it's just the shell here. Yeah, so. A question again, maybe it's just for reflective light viewing. Yes. Yeah, with exactly the 4 times that I have with top illumination, I need a strong flashlight, which I don't have here right now. Then we're able to see it also much better. Okay, so people, I'm already, people are saying here, sounds as if you have a little flu or well, I've got the common cold virus, so-called a rhinovirus it's called. I usually get a common cold around 2 or 3 times a year. And the reason why I've got a cold now is because last week I was not home. I actually went on a marine biology week with a whole bunch of students of mine. And the water was pretty cold, 13 degrees centigrade. So I think that might have contributed a little bit to my cold. But why am I telling that to you? I'm telling that to you because I'm preparing a video of this, because I was actually able to find a couple of interesting plankton samples that I also put under the microscope as well. And so I'm going to make videos of this that I'm also going to publish on this channel here. Okay, this explains a little bit the reason why I've got a cold right now. Yeah. Mobile phone flashlight would work, but is not strong enough. Okay, I tried that. I need a much stronger flashlight. So I'm going to call it quits for today, people. Hope that you liked it. Okay, for those of you who joined in late, a very, very brief summary. So to give you a little context, I bought some antique microscope slides, and I just showed them to you. And if you want to also have a look at the first set of slides here, go back into the archive on February 7th. I also did a live stream where I showed you the first set of slides as well. Okay. Yeah, I wish you all the best. See you around next time. Hope to see you again next week. And until that time, happy microbe hunting. Okay. And bye-bye.